Hello and welcome to Science Research Weekly, Season 4, Episode 2, Antivenom from Scratch. Get ready for long-lasting DNA lesions, DNA nanorafts, de novo antivenom, a second look at SAS blog posts, working with colors in R, and K-type awards from NIH. This episode covers material from January 10th to the 16th. Let's get started with some research articles. First up here, prolonged persistence of mutagenic DNA lesions in somatic cells. Researchers looked at mutations for DNA lesions across human cells from donors and found that lesions from both endogenous and exogenous mutagens can persist for long periods of time, months, or even years. So here's a example kind of different pathways. They kind of use a sort of, I guess, no, I think they actually called it phylogenetic analysis. So you can see kind of the phylogenetic as you were history of different lesions across replication. All right, next up. Stretching and immobilizing a single DNA molecule on a glass surface using pressure flow in a microchannel for super resolution imaging. So researchers improved an already existing single DNA analysis by stretching out the DNA molecule flat and keeping it immobile. And so you could get better resolution. I think a really helpful picture here is kind of this here we see on the right side, the schematic and how it doesn't look very clear when it's coiled up, but if you stretch it and immobilize it, it looks nice and straight. I think there's better pictures of sort of seeing them all in various microchannels. Okay, moving along. Our next article is Morphology, Remodeling, and Membrane Channel Formation in Synthetic Cells via Reconfigurable DNA Nanorafts. So researchers created synthetic cells, or maybe more accurately cell-like objects, called giant unilaminar vesicles, or GUVs. They were able to reshape them using DNA nanorafts, showing a use of DNA nanotechnology, so folding DNA into useful shapes like origami, to modify synthetic cells. Let's see if we can find some good figures here. So here's sort of like the schematic of these synthetic cells and how they can be sort of, the membranes can be deformed and changed across time here. Okay. Our next research article is an evolutionarily conserved metabolite inhibits biofilm formation in E. coli K12. So biofilms, those are collections of microbes, usually bacteria, that stick together on a surface, causing a lot of issues. Researchers found that the chemical methyl atriethyl cyclodiphosphate, or MECPP, which is a stress signal in plants, can inhibit biofilm development. So helpful to have a natural product from plants that we can use to combat biofilms. I know biofilms in space are a big problem, so maybe we can get these plants up there, or these plant chemicals, to deal with things like that. So, all right, rounding out our articles here, probably the most interesting, de novo designed proteins neutralize lethal snake venom toxins. So snake venom, usually treated with costly antibodies with limited efficacy, especially across this certain type of toxin, this 3FXT. It's the three-finger toxins. These are from, what is it? Elapid snakes. But using deep learning, researchers designed proteins that actually bind to the three-finger toxins with good results, even protecting mice from an otherwise lethal neurotoxin dose. I think here's some sort of conceptual designs of like the toxins and how they'll sort of bind to it. But then I think the real kicker of the figures is going to be here, where it shows Things like survival rate of mice, controls, they all die, but if you have different, even shorter long binders, you can see that most of the mice survive. 1 to 10, 1 to 5, toxin to binder. So doing a lot to keep these mice alive. All right, let's switch to some research tools. Last week, we went over Rick Wickland's top do-loop blog posts from 2024. Now he's back with 12 blog posts that deserve a second look. Under statistics and data analysis, there's the use the moment ratio diagram to visualize the sampling distribution of skewness and kurtosis. There's the distribution of p-values under the null hypothesis. And there's using flexible distributions to fit data, which I really like. That's, as he calls it, the holy grail of statistical modeling is to find a flexible distribution that can model any continuous univariate data. On the regression side, you have 3 posts: Poisson regression in SAS, visualize a multivariate regression model, and isotonic regression. So they might be of interest if you're regression heavy. Moving along, there's simulation and bootstrapping articles, distribution of the R-squared statistic, simulate correlated continuous and discrete variables, and introducing proc-sim system. Rounding out, he has 3 final articles he thinks you might want to take a second look on. Under the topic of statistical graphics, there's the correllogram, visualize correlations by fitting angles. There's also scale a density curve to match a histogram, and finally, create filled density plots. There's an interesting new R bloggers post from Nicola Rennie, working with colors in R. It involves different ways to define colors, choosing colors, and even generating colors yourself, making a custom-made color palette. On the crayon side of things, there's new crayon packages of note. 7, if you're keeping track at home. There is part tree, visualize simple 2D decision tree partitions. RW life table, estimate of life tables using rolling windows. Prot-E, processing proteomics data, statistical analysis and visualization. PS power, sample size calculation for propensity score analysis. GG stack plot, create overlapping stacked plots. HHMR, hierarchical heat maps, and BNNS, Bayesian neural network with STAN. Finally, on our research funding side of things, 2 new grant opportunities. First from the NCI, National Cancer Institute, Pathway to Independence Award. This is a K99/R00 with a clinical trial required. This program is to facilitate a timely transition of talented postdocs to research or clinical doctorate degree, or from them to a postdoctoral position to independence. So this is a pathway to independence. Maybe I should just said that, or you could have just read the title. All right, second, you've already got a K-type award. There is from the National Institute of Diabetes and Digestive and Kidney Diseases. There's a small grant program for different K awards. This is technically an RO3, clinical trial optional, helping those award recipients conduct research as they complete their transition to fully independent investigator status. Snakebite, schmakebite, thanks for joining me for Science Research Weekly. Science on.